Hello, everyone. Welcome back to Science in the Age of COVID-19 seminar series. We are back after a short break last week. In course of this pandemic, we have been learning a lot about this virus, the mechanism by which it interacts with our system to cause the infection and the after effects of the infection. Today, we'll hear about the possible mechanism behind the neurological symptoms that have been observed in patients infected with this virus. It's our pleasure to host Dr. Chris Bartley, who is a Hanna H. Gray Fellow and a postdoctoral fellow at UCSF. And today, Chris will talk to us about his work on the possible role of autoantibodies in COVID-related neuropsychiatric disorders. Chris, thanks for your time, and we look forward to hearing from you.
Hi, thank you for the kind introduction, and I'd first like to thank HHMI, Janelia, and Loren Looger for inviting me to speak to you today. It's really an honor to be here speaking to this group, and as you mentioned, I'm a Hanna H. Gray Fellow. I'm doing a postdoctoral fellowship with Sam Pleasure, who's a neurologist and a developmental neuroscientist at UCSF, as well as with Michael Wilson, who's a neurologist focused on advancing diagnostics for neuroinflammatory diseases. And I'm a physician scientist with a training in psychiatry. And my interest is in this growing field of immunopsychiatry and whether or not autoimmunity may cause psychiatric symptoms or neuropsychiatric symptoms in subsets of individuals. Let me go to the next slide. So I have no disclosures. And just a little bit of an outline for the talk today. So first I'll talk about the background of neuro-COVID, and then I'll move into some of the data that we have for anti-SARS CoV-2 and autoreactive antibodies in bulk CSF of patients with neuro-COVID. Then we'll move on and talk a little bit about immunoreactivities of a small handful of monoclonal antibodies that we've derived from CSF expanded B cells from one patient with COVID-19 and neurologic symptoms, and maybe discuss a little bit about some of the evidence for molecular mimicry. And then we'll move on to talk a little bit about preliminary findings from our expanded neuro-COVID cohort, and these are unpublished data. And then finally, I'll talk about conclusions and next steps. And I'm going to start a little bit away from COVID-19 to begin with. When I applied to be a Hanna H. Gray Fellow, my application was focused around autoantibody discovery in psychiatric populations, in particular in psychotic spectrum disorders. And during residency, and I was particularly motivated by some emerging literature that showed that there was a epidemiologic relationship between autoimmunity, infections, and a future diagnosis of schizophrenia. And so this is just a study that came out of Denmark, where they have an integrated EMR, and they looked at the entire population and asked whether or not a history of severe infection was associated with an increased risk for a future diagnosis of schizophrenia, and that's the green line. And the answer was yes. The more times someone had been hospitalized with severe infection, the more likely they were to be diagnosed with schizophrenia. They also asked whether this was true for individuals with autoimmune disorders, and the answer was yes. And when they looked at the intersection between those two, they found that those individuals who had a prior infection, severe infection, and a diagnosis of a systemic autoimmune disorder were at an even greater risk of a future diagnosis of schizophrenia. And so, these data really suggest that perhaps there's a relationship between the immune system and neuropsychiatric disorders. Now, the mechanism is entirely unclear, but COVID-19 may offer us an opportunity to start to test this hypothesis or this relationship. And so, like many of you, my research was put on hold because of COVID-19, but many of the molecular tools that we were using to answer this question or to study this question in the psychiatric population, we were able to pivot and direct towards the COVID-19 population. And I was very I was very pessimistic that we would find anything, because COVID-19 is obviously due to a virus that infects the pulmonary system. But as many of us soon found out, COVID-19 is a pulmonary disease with extrapulmonary manifestations. And so on the left, I'm showing you data that came out of an early study that was published in JAMA Neurology, where they found that 30% of patients with moderate to severe COVID-19 went on to develop neurologic complications that included seizures, anosmia, ageusia, hemorrhagic encephalitis, delirium, and encephalopathy or global brain dysfunction. And that's a short list of some of the neurologic complications that COVID-19 patients can suffer from. And then subsequently, we found that there's evidence for substantial neurologic and psychiatric morbidity in the 6 months after COVID-19 infection. And we found that this was a study that came out of Paul Harrison's group, and they found that there was an elevated incidence of new or recurrent mood disorders, psychotic disorders, anxiety disorders, and substance use disorders. And this study that came out of Harrison's group was really important because they didn't look at the increased incidence relative to a healthy population. They actually looked through 81 million electronic medical records of COVID-19 patients and other patients in the United States, and compared COVID-19 patients to those who had had a prior respiratory tract infection. And then they did a secondary comparison to those individuals who had had an influenza infection. And they found that the incidence of neurologic and psychiatric disorders was increased in those individuals with COVID-19 relative to those other two populations with the prior respiratory tract infection. So what is the mechanism behind this relationship between neurologic impairment, psychiatric symptoms, and COVID-19? There have been many hypotheses, and the reality is there's probably not a single mechanism. And so we're looking for multiple mechanisms, and today I will talk about autoimmunity. But some of the leading early hypothesized mechanisms included neuroinvasion. However, RT-PCR, cerebrospinal fluid and brain parenchymal tissue, which I'll show you in a moment, was often negative. There was some question as to whether or not systemic inflammation may also reflect central inflammation, whether or not cytokine storm or elevated CSF, cerebrospinal fluid cytokines, may contribute to global brain dysfunction. And Carlos Pardo at Johns Hopkins University has done a really nice study where he's looked at cerebrospinal fluid of neuro-COVID patients relative to patients with other neurologic problems, and has found that this cytokine storm hypothesis doesn't seem to explain the neurologic impairment that we see in these patients. Thromboses, and although there is an increased risk of blood clots or thromboses in patients with neuro-COVID, and a small increased risk in some individuals who may be vaccinated with the adenovirus-based vaccines, this doesn't fully explain the neurologic complications that we see. And then toxic metabolic encephalopathy, which is sort of a catch-all term that describes global brain dysfunction in the setting of metabolic dysfunction. And then finally, there's autoimmunity, which I'll talk to you about today. Before I get into autoimmunity, I just wanted to review some of the data around evidence for SARS-CoV-2 neuroinvasion. And this is from a recent paper that was published in Brain, where they performed RT-PCR on different levels of the neuraxis, starting at the nasopharyngeal cavity. And they performed RT-PCR for both the S and N genes of SARS-CoV-2 on nasopharyngeal swabs, nasal epithelium, olfactory bulb, superior frontal gyrus. You can see the matrix there on the left. And what they found was that although on hospital admission for these patients who ultimately went on to pass due to COVID, they were able to detect a high burden of SARS-CoV-2 RNA based upon a low cycle threshold number. As you moved into brain parenchyma, SARS-CoV-2 RNA was either undetectable or had a very high cycle threshold number. And importantly, there was a letter that was published in NEJM recently that found in serial nasopharyngeal swab samples from hospitalized patients that when the cycle threshold was over 28, they were unable to culture virus from nasopharyngeal swab samples. And as you can see here, within the brain parenchyma, there are no patients who have RNA at a cycle threshold above or lower than 28, suggesting that although you might be able to detect RNA here, it's likely not culturable. On the right, they use RNA scope to try to detect the anatomic distribution of any potential SARS-CoV-2 RNA within brain parenchyma. And you're looking at two patients. The first column is patient 6, and the second column is patient 5. And they're looking in the medulla oblongata, and they were unable to detect either RNA probes binding to end transcripts or spike transcripts, whereas they were able to detect SARS-CoV-2 RNA in the lungs of these patients shown on the right. So the question as to whether or not SARS-CoV-2 is neuroinvasive is outstanding. It may be in some rare instances, but the question of whether or not it's neuroinvasive to a meaningful degree is not clear. At the same time, we see that there is a lot of evidence for systemic humoral autoimmunity in COVID-19. And so there are a number of papers that have demonstrated this, and I'm just highlighting three here. There's a paper by Ignacio Sanz's group out of Emory that showed that there was extrafollicular B cell activation that was similar to what they've seen in lupus, where you get this clonal expansion of B cells at extrafollicular sites that can lead to antibody production that's diverse and low affinity but autoreactive. And then out of Casanova's group, many of you are probably familiar with this, but there's evidence to suggest that there's maybe 10% of males with severe COVID-19 harbor these anti-interferon alpha or omega antibodies, these type 1 interferon autoantibodies. These autoantibodies are functional and interfere with the immune response. And then Aaron Ring's group, using this rapid extracellular antigen profiling technology that he's developed, it's a yeast display type of technology, has found that there are diverse functional autoantibodies that often target components of the immune system that are present in the serum of many patients with COVID-19. Now, what these studies don't answer is whether or not these autoantibodies were a sequelae of COVID-19 or predate COVID-19 and therefore sculpt the immune system's response to COVID-19. There's a small, I would say, underappreciated study that came out of Spain that has demonstrated post-COVID autoimmune seroconversion. And here they looked at serum samples from patients with severe COVID-19, some with neurologic impairment, and stained rat, liver, and gastric mucosae with the serum from these patients. And some of these patients had blood samples that had been biobanked during the pre-pandemic period. And they were able to demonstrate that a subset of these patients develop autoantibodies that bind to gastric and liver mucosae in the post-infectious period, which provides stronger evidence for COVID-19 eliciting an autoimmune response. So the relationship between viral infection and autoimmunity is well established. Viral infections are often associated with autoimmune system syndromes of the nervous system. In the peripheral nervous system, influenza and cytomegalovirus have been associated with Guillain-Barré. And then in the central nervous system, herpes simplex virus, Japanese encephalitis virus, and varicella zoster have also been associated with the onset of autoimmune encephalitides, most commonly anti-NMDA receptor encephalitis. And this has now been shown to be associated with SARS-CoV-2 in rare cases as well. And then on the neuropsychiatric spectrum, it's well known that influenza, either a wild type infection or even in some cases an influenza vaccine, can lead to narcolepsy. And narcolepsy is caused in these cases by a loss of orexin-producing neurons in the hypothalamus. So despite this relationship between viral infection and autoimmunity, when we look at COVID-19, the CSF from neuropsychiatric COVID-19 patients is rarely inflammatory. So there's not much evidence at the clinical level of inflammation within the CNS. SARS-CoV-2 RNA, as I mentioned before, is rarely detected in the CSF, but it's also rarely detected in the brain parenchyma. And the traditional autoantibody testing is generally negative in neuropsychiatric COVID-19 CSF biospecimens. And so we wanted to ask whether or not, using more unbiased approaches, we could find a signature for autoimmunity in the cerebrospinal fluid of patients with neuro-COVID. And so this data has been published, and some of you may have seen the paper that came out in Cell Reports Medicine. This is a collaboration between our group and Shelley Farhadian's group at Yale University. Shelley Farhadian's a wonderful infectious diseases doctor. And she initially put together a cohort of 7 COVID-19 patients whose ages ranged from 16 to 84, and there were 5 males and 2 females in this group, and all of these patients had moderate to severe COVID-19. And all patients were positive for SARS-CoV-2 by nasopharyngeal swab. However, none of these 7 patients were positive for SARS-CoV-2 in their cerebrospinal fluid. And then the neurologic symptoms were diverse, as indicated there. And many of these patients, as we've seen in patients with COVID-19 that's severe or moderate, many of our patients had a number of comorbidities. And so Shelley Farhadian's group initially looked at the transcriptional profile of lymphocytes in the CSF and the periphery of COVID-19 patients. And by flow cytometry, they were able to detect an increased frequency of B cells, these are antibody producing cells, in the cerebrospinal fluid of these patients, and that's shown in the lower right here. And so this suggested that perhaps there was some sort of humoral immune response in the cerebrospinal fluid. Now, as I mentioned before, SARS-CoV-2 RNA is rarely detected in the CNS, and so we wanted to ask whether or not SARS-CoV-2 neuroinvasion is required for an intrathecal humoral response, intrathecal meaning within the cerebrospinal fluid. And we can't ask this question in humans, and so we wanted to ask whether it's required in mice, and therefore may also be true in humans, although we don't know. So one of our collaborators at Yale School of Medicine, this is Eric Song, who's a really talented MD PhD student out of Akiko Iwasaka's group, used an adeno-associated virus to express the ACE2 receptor for SARS-CoV-2 in the brain, the lungs, or in both organs. And this allowed us to reliably dissociate pulmonary neurologic infection of SARS-CoV-2. And so after direct infection of either organ, we detected SARS-CoV-2 in the RNA in the lung and the brain, and this was short-lived. However, similar to clinical findings, we did not detect SARS-CoV-2 RNA in the CSF of these mice. So when SARS-CoV-2 is administered intranasally to mice expressing the human ACE2 receptor only in the lungs, thereby causing pulmonary but not brain infection, we detected significantly elevated anti-spake SARS-CoV-2 antibodies in the lung and the serum, but not in the brain or the CSF. So when SARS-CoV-2 was administered intranasally to mice expressing the human ACE2 receptor in the brain and the lungs, thereby leading to pulmonary and brain infection, we detected anti-spike antibodies in all 4 compartments, the lung, serum, brain, and the CSF. And then finally, when the human ACE2 receptor was expressed in the brain only and SARS-CoV-2 was administered intracranially, thereby causing infection only in the brain but not in the lungs, we detected anti-spike antibodies in the brain and in the CSF, but not in the lung or the serum. And so this really suggested that CNS SARS-CoV-2 infection is sufficient to elicit a humoral response in the cerebrospinal fluid. We don't know whether it's required, though. So given the increased frequency of B cells in the cerebrospinal fluid and this animal data that's suggesting the possibility of a compartmentalized humoral immune response in individuals with SARS-CoV-2 infection, we wanted to do a broader profiling of humoral immunity in the cerebrospinal fluid of these samples. And so we use 2 basic techniques. The first technique is just mouse brain immunostaining. And so this looks for anti-neural autoreactive autoantibodies. And this is a classic and traditional and standard method for searching or for screening for autoantibodies that are reactive to neural tissue. Mouse brain proteins are about 85% similar to human proteins at the amino acid level. And so if an antibody binds to mouse brain, we presume that it binds to human brain as well. This technique is actually used clinically to diagnose some types of autoimmune encephalitis. And so we take 12 micron sections of sagittal mouse brain and incubate them with cerebrospinal fluid. In this case, we did it at a 1 to 10 dilution. Clinically, these samples are screened at a 1 to 2 dilution, so this was more stringent. And then we wash those brain sections, we counterstain for human antibodies using an anti-human IgG antibody, and then use an axioscan to take panoramic images to look at the anatomic distribution of any antibodies that have bound to brain tissue. And then the next thing we do is so this tells us whether or not there is an antibody that's capable of binding to neural tissue, but we also want to know what the direct targets of these antibodies are. And so we use a technique called T7 phase display immunoprecipitation sequencing. And so our approach is really inspired by the original development of the FIPSeq platform by HHMI investigator Stephen Elledge and his mentee Ben Larman at the time, and they're still really evolving this approach and generating new and exciting data each day. But we've designed our own FIPSeq platform, and so I'm going to explain to you how our human peptidome is designed. So all known proteins and protein variants were bioinformatically divided into 49 amino acid peptides that overlap by 25 amino acids. And then we printed oligonucleotides encoding these peptides and cloned them into T7 bacteriophage. And each of these bacteriophages then displays a single peptide on its outer surface. And in our instantiation of the phage display platform, each individual T7 bacteriophage displays about 5 to 15 copies of an individual 49 amino acid peptide. And so it takes about 700,000 unique phage to tile the entire human proteome. And we call this comprehensive set of phage a library. So we incubate this library with blood or cerebrospinal fluid, and antibodies are allowed to bind to their peptides. If they do bind to human peptides, we wash away the phage that aren't bound by the antibodies, and after the antibodies have been absorbed to magnetic beads. And then we elute off the antibodies in the bound phage, and we sequence those phage. And then we map the sequence peptides back to our reference proteome or peptidome to get a list of candidate targets. So for this project, we screened the biospecimens on our human peptidome, but we also screened them on a pan-virome, which is encoded with peptides that tile all viruses that are known to infect mammals, as well as a dedicated seasonal human coronavirus phage display platform, as well as an even more targeted SARS-CoV-1, SARS-CoV-2 phage display platform. And the characteristics, i.e. the length of the peptides, the degree of overlap, and the number of individual phage, varies across each of these libraries. So Colin Zemechnik, who is a really talented bioengineer, shared between the Wilson labs and the Derisi labs here at UCSF, and then Jayant Rajan, who's an MD PhD infectious disease doctor, worked together to build the dedicated seasonal coronavirus phage display platforms, as well as the more targeted SARS-CoV-2, SARS-CoV-1 coronavirus phage display platforms. And they use these platforms as well as our pan-virome to evaluate 95 or 98 confirmed SARS-CoV-2 biospecimens that had been collected. These are blood samples that had been collected from patients with clinically validated SARS-CoV-2 infection that had been validated by either RT-PCR or a nasopharyngeal swab, or a 50% neutralizing plasma antibody titer. And so they compared the enrichments of various peptides on the phage platform to 95 de-identified pre-pandemic adult control sera that we had obtained from the New York Blood Center. And they looked for antigens that were specifically enriched by individuals with a confirmed SARS-CoV-2 infection to develop a platform that I'm going to tell you about in a moment. This work was published in Cell Reports Medicine, and there's a new technique for autoantibody discovery that Colin developed called Rescan, and so I really recommend that you guys check it out if you haven't had a chance to read it. In any event, using this approach where they look for differentially enriched peptides by individuals with SARS-CoV-2 infection, they were able to identify 4 peptides mapping to SARS-CoV-2 spike protein and 4 peptides mapping to the the SARS-CoV-2 nucleocapsid protein that discriminated between bona fide SARS-CoV-2 infection and, say, seasonal coronavirus exposure. And so on the left, I'm showing you where those peptides map onto the spike protein. And you can see that the receptor binding domain is shown in black here, and it's in the up conformation. And then these 4 different peptides are shown beneath that. One is in red, which is just at the C-terminal portion of the receptor binding domain. It's a little bit difficult to tell, but there's just beneath that, there's a dark green splotch, which is even further C-terminal. It's a little bit N-terminal to the fusion peptide. There's a bright green spot which is also just a little bit N-terminal diffusion peptide, and then we have this very C-terminal peptide on the spike protein. And then 2 of the 4 nucleocapsid peptides that discriminate between SARS-CoV-2 infection and non-infected status are shown here, and the other 2 are out of view. So using these peptides, as well as the whole spike protein, the receptor binding domain, the nucleocapsid protein, as well as one peptide mapping to open reading frame 3A, Colin created a Luminex-based antigen panel to profile for SARS-CoV-2 antibodies and biofluids from individuals. And so we had sufficient biospecimen to pan the plasma of 5 patients initially in this initial cohort, and the CSF of 6 of those patients. And what we found is that SARS-CoV-2 antibodies were detected in the plasma of all 5 patients. And surprisingly, SARS-CoV-2 antibodies were detected in all 6 of patients as well, in the CSF of all 6 patients. And so this, really sort of mirrors a little bit indirectly, but mirrors some of the animal data in which we were able to detect anti-spike antibodies in the cerebrospinal fluid of mice that had been artificially intracranially infected with SARS-CoV-2. And so we wanted to know what else was lurking in the bulk CSF of these biospecimens. These patients uniformly had anti-SARS-CoV-2 antibodies, but might they also have autoreactive antibodies? And on the left, I'm showing you a binary matrix of the immunostaining results that we got after staining rodent brain tissue in a 1 to 10 dilution. And we found that 5 of 7 of the biospecimens stained at this dilution. 3 of them went on to stain at a 1 to 50 dilution, and 4 of them stained at a 1 to 25 dilution. We also stained with control CSF samples, and only 2 of the 6 samples that we screened immunostained rodent brain tissue at a 1 to 10 dilution, and none stained at more dilute concentrations. And those 2 that did stain had a pretty nondescript and weak immunostaining pattern. And on the right, I'm showing some examples of immunostaining that was produced. And we saw that there was a shared immunostaining pattern amongst 3 cases that localized the CA3 of the hippocampus, as well as the hilus and the dentate gyrus. And this immunostaining pattern has been reported by some other individuals out of Europe as well. We found that there was one patient who had immunostaining that localized to endothelia within the mouse brain cortex. We also found examples of immunostaining in case 3 just beneath that of cells that line, these look like pericytes or cells that line large vessels. So we wanted to ask whether or not, so we knew that there were antibodies that bind to SARS-CoV-2. We also found that there are antibodies that bind to mouse brain tissue. We were curious to know whether or not there were antibodies that bind, what these antibodies bind to. So we ran our samples on the phage display immunoprecipitation sequencing platform, and I'm only showing 2 examples here, but as a group, we found that these samples enriched many more peptides and control cerebrospinal fluid. We also found this by immunoprecipitation mass spectrometry. However, I'm not going to talk about that data today. And one of the antigens, the candidate antigens that we identified by phage display, was this intraflagellar transport protein 88, IFT88. On the left, I'm showing you that 2 peptides that overlap each row in this heat map represent one peptide, and these 2 peptides are neighbors, and they share an epitope between them. Beneath case 1, I'm showing 2 replicates. And so we saw that these 2 peptides were enriched in both replicates of the CSF from this patient, but not the plasma. And we did not see enrichment in the other COVID-19 patients or in the controls. And in those other columns, those are the mean values of the technical replicates for COVID-19 and control patients. And so we wanted to ask whether or not this candidate autoantigen that was predicted by phage display indeed was a bona fide autoantigen. And so we transfected an RFP tag IFT88 protein into HEC293 T cells, fixed those cells and immunostained them with cerebrospinal fluid with an antibody to RFP, as well as a commercial antibody to IFT88. And we found that this autoantibody was indeed a valid autoantibody. Interestingly enough, mice that have deletions in IFT88 go on to develop anosmia, or inability to smell, and humans that have mutations in this gene develop ciliopathies. And so this is a protein that's highly enriched in the cilia of the pulmonary epithelia, as well as cilia that are expressed in the brain. And then we validated another protein that's poorly characterized called FAP3. And even though it was detectable in the plasma, the enrichment scores were greater in the cerebrospinal fluid of case 3. We validated this autoantibody by Western blot. And the function of this protein is not well known. Both of these proteins are intracellular, and so it's not clear whether or not these autoantibodies are functional. So that's an example of 2 autoantibodies that we validated, we identified by phage. We have some others that we haven't disclosed yet. But one of the problems is that these antineural antibodies could have originated from the periphery. Just because we detect them in cerebrospinal fluid doesn't necessarily mean that they were produced by an inflammatory response in the central nervous system. And so we looked for additional evidence of humoral response within the CSF. So in response to a target, antigen B cells will clonally proliferate. And in one patient, we identified multiple clonal populations of B cells in the CSF that were not detected in the periphery. And this was by 10x sequencing, and the sequencing depth could have been deeper. So just because they were not detected in the periphery doesn't mean they weren't present in the periphery. Nonetheless, these were, based on our sequencing depth, uniquely expanded in the cerebrospinal fluid. And so this suggested that perhaps these B cells were responding to an antigen in the CNS. So we cloned monoclonal antibodies from the 5 most abundant clonally expanded CSF unique populations, as well as a representative B cell from peripherally unique clonal populations. And then the frequency of those BCR, this B cell receptor clones, is shown on the right. And so we wanted to ask, what do the monoclonal antibodies derived from CSF expanded B cells bind to? And we found that some of these antibodies bind to the spike protein with high affinity. So we used our same Luminex-based platform to screen for reactivity to SARS CoV-2 antigens. And we found that one of the CSF-derived monoclonals, mAbs-C2, bound to whole spike protein, and that 2 of the peripherally derived monoclonals, P1 and P2, bound to whole spike protein. And then none of the monoclonals bound to any of the other SARS-CoV-2 antigens represented on this Luminex platform. You'll see that P4 is missing, and initially that antibody did not express well in culture, so we weren't able to screen it. So we found that they bind to whole, a subset of these monoclonal antibodies bind to whole spike protein, but we wanted to characterize them a little bit further. And so we turned to Wesley Wu and John Pack in the BioHub located here at UCSF, the Chan Zuckerberg BioHub. And they used a 2P, a 2-protein stabilized spike protomer to determine the binding affinities of our mAbs using biolayer interferometry. And so they generated binding curves for each antibody at the spike protomer concentrations ranging from 0 to 316 nanomolars. And as a positive control, they used antibody CR3022, which originally derived from a SARS-CoV-1 patient, but has been shown to bind tightly to SARS-CoV-2 spike protein. And they found that all 3 of our monoclonal antibodies, mAbs-C2, P1, and P2, bind with relatively high affinity between 0.2 nanomolar for P1 and 2 nanomolar for mAbs-C2 and P2. And so these KDs are within the range of some published neutralizing antibodies. So we wanted to ask whether or not these antibodies do in fact neutralize SARS-CoV-2. And so with help from Sarah Sunshine, who's a graduate student at the Derisi lab, we screened for neutralizing activity against wild type SARS-CoV-2 from a strain that had been isolated from a patient here at UCSF. And so we pre-incubated this clinical isolate of SARS-CoV-2 with our monoclonal antibodies and a mouse monoclonal positive control antibody targeting the spike protein at concentrations ranging from 2.5 to 25 micrograms per ml. And after co-incubation of the antibody with the virus, we infected VROE6 cells or HUH cells that overexpress H2 and the TMPRSS2 protease at an MOI of 1. And the following day, we fixed and stained these cells, and we stained for the nucleocapsid protein. And what you can see here is that SARS-CoV-2 nucleocapsid protein is readily detectable when SARS-CoV-2 is used to infect these 2 cell lines without mAb or monoclonal antibody pre-incubation. If you pre-incubate with the neutralizing control antibody, you substantially reduce or eliminate the nucleocapsid detectability, indicating the virus has not infected these cells. However, even up to concentrations of 25 micrograms per ml, mAbs-C2, P1, and P2 were not able to interfere with the infectability of SARS CoV-2. So these antibodies are not neutralizing, and perhaps this is not that surprising because based on the Luminex panel, we found that these antibodies don't bind to the receptor binding domain. And most neutralizing antibodies appear to bind to the receptor binding domain, although there have been some that are reported to bind outside of this region. So what do the other CSF-derived monoclonal antibodies bind to? They're clonally expanded, but what do they bind to? And so we turned back to mouse brain tissue staining, and once again did panoramic images of immunostained mouse brain sections that had been incubated with each of our monoclonal antibodies from the periphery and the cerebrospinal fluid. And as a negative control, we used a monoclonal antibody targeting the hemagglutinin antigen in influenza that was in the same IgG1 backbone as our SARS-CoV-2 patient-derived monoclonal antibodies. And we found that none of the peripherally derived monoclonal antibodies were autoreacting to mouse brain tissue. And a representative example is mAbP7, shown in the middle on the left here. However, 4 or 5 of the brain-derived monoclonal antibodies were immunoreactive to mouse brain tissue. And I'm showing an example, mAbC4 here on the bottom. On the right, I'm showing examples of different immunostaining patterns that were produced by these monoclonal antibodies. And importantly, mAbC2 is also our anti-spike monoclonal antibody. And so this suggests that perhaps there are some anti-spike antibodies that can react with rodent brain tissue and perhaps with human neural antigens as well. And this immunostaining was done with help from Thomas No, who's a really talented technician in the lab, as well as Trung, who's our lab manager. And this is just showing a closer view, a more close-up view of the immunostain that was produced by mAbC2. You can see that this is taken from the neocortex on the left and the hippocampus on the right. And you can see that there is some somatic staining and some cytoplasmic staining, but also you can see that this arrow is pointing to what looks like neuropil, and there's this sort of this diffuse immunostaining of the tissue produced by mAbC2, both in the cortex and in the hippocampus. And when we see this pattern, we start to think that perhaps the antigen targeted by this antibody is extracellular. Extracellular epitopes tend to produce this more diffuse immunostaining pattern, and extracellular antibodies that target extracellular antigens are more likely to be functional. So we wanted to characterize mAbC2 a little bit more closely, and so we paired up with Christopher Barnes, who's a phenomenal structural biologist. Who's I encourage you to have him speak here if he hasn't. He's also a Hanna H. Gray Fellow who's going to be starting his lab at Stanford soon. And he developed a pipeline for interrogating the sort of structural for interrogating antibody binding to the spike protein, and ran our monoclonal antibody through this pipeline. And typically uses the 6P stabilized trimeric spike protein, but in our case, the antibody was only able to bind to the 6P stabilized monomer. And based on some of the EM micrographs, it looks like this monoclonal antibody may actively dissociate the spike trimer protein, although we don't know that for sure. And so the preliminary modeling really suggests that this patient's monoclonal antibody targets loops. There's 2 discontinuous loops at the N-terminal portion of the spike protein. N-terminal domain loop 37 to 48, those are the amino acids, as well as amino acids 280 to 285. And then at 282, there's an N-linked glycan that's relatively large that also participates in antibody binding, and that's not visualized here. Those 2 discontinuous loops are shown in this in the ribbon diagram within the circle to the right here. And so this glycan, I think, is somewhat interesting because many proteins that are secreted in the brain are heavily glycosylated, and so it's pretty weak evidence, but it's sort of consistent with the fact that perhaps this antibody binds to an extracellular antigen within the brain as well. So those studies are ongoing. So what about these other mAbs? We found that some of the monoclonal antibodies recapitulate the immunostain that's produced by bulk CSF IgG, and each of these pairs, left and right, shows an example of bulk CSF immunostaining of mouse brain tissue, and then, of course, the most similar immunostaining pattern that was produced by the monoclonal antibody. And this suggests that in some cases, these monoclonal antibodies may actually reflect, IgG populations that are relatively abundant in bulk CSF. So I mentioned that one of our monoclonal antibodies binds to spike protein, but we actually found that two antibodies bind to spike protein. Although mAbs-C4 was negative on the Luminex panel, we also ran these monoclonal antibodies on our dedicated SARS CoV-1, SARS CoV-2 peptidome. And we found that there were 5 overlapping peptides that map to the spike protein that were enriched by mAbs-C4 more so than the influenza control antibody that was also run on the same platform, the anti-hemagglutinin antibody shown at the bottom here. And so we asked where these peptides map to on the spike protein, and we found that they map to a region that's just N-terminal to the fusion peptide, and the sequence, multiple sequence alignment for these 5 peptides that map to this region is shown here. And if you map these onto the reference proteome for the spike protein, you find that they fall between amino acids 780 and 798. So mAbs-C4 was one of these. So we wanted to validate that this monoclonal antibody does indeed bind to the spike protein. It was enriched by coronaphage or by SARS-CoV-2, SARS-CoV-1 phage, but didn't bind to the whole spike protein, the Luminex, and so acquired recombinant S1 protein or recombinant spike extracellular domain protein, which contains the S1 and S2 subunits. And that sequence that was enriched by the SARS-CoV-1-2 peptidome maps to the S2 subunit. And so I ran those out by SDS page. I loaded about 0.5 micrograms into each lane and then probed with 0.25 micrograms per ml with monoclonal C4. And we see that the immunoreactivity is specific to the S2 containing subunit, but not S1. And then the anti-6S His immunostaining is here, just showing that equal amounts of protein were loaded into either lane. And so this really validated that mAbs-C2 is an anti-spike protein, but it likely binds to an epitope that's cryptic in the whole spike protein. mAbs-C2 also bound to mouse brain protein, and so this is potentially another cross-reactive monoclonal antibody. And we wanted to know, what the target within the brain was. And so we ran mAbs-C4 on our human peptidome, and the heat map of the peptides that were enriched relative to the influenza controls antibodies and beads is shown on the left. And you'll see 4 arrows highlighted in green, and those map to 4 different proteins that were enriched above our threshold by on the human peptidome, and also enriched by immunoprecipitation mass spectrometry. And this graph on the right is pretty preliminary and hard to read, but you can see the 4 proteins on listed below. And we found that there were 2 proteins, PDE4-DIP and FIP1L1, that were uniquely detected in the IP from mAbs-C4 and not detected in the IP from any of the other mAbs or control antibodies. And so after screening for immunoreactivity to those 4 proteins, one of them validated. And so we transfected HEC293 cells with a flag tagged FIP1L1 construct, and then immunostained with the antibody at 1 microgram per ml, not shown here, 5 micrograms or 10 micrograms per ml, and found that the immunostain pattern perfectly co-localized with the anti-flag pattern, and that this monoclonal antibody does not stain untransfected HEC293 cells. The 1 microgram per ml condition also worked. And as a secondary validation, I overexpressed these proteins in HEC293 cells and ran out the proteins by SDS page, along with a flag tagged CDR2 construct. This is an unrelated protein, to ensure that the antibody wasn't non-specifically binding to the flag tag. And so you can see that in both of these assays, the monoclonal antibody binds to FIP1L1. FIP1L1 is an intracellular protein. It's involved in the polyadenylation complex. And so it's intracellular, it's not specific to the brain. And so even though this antibody seems to cross-react with spike in FIP1L1, we're not sure that it's functional. So as I get to the end, I just would be remiss if I didn't circle back to where I started, which was to think about this relationship between infections, autoimmunity, and pure psychiatric disorders. So we've been collecting biospecimens from neuro-COVID patients throughout the pandemic. And some of these patients have presented with isolated psychosis, which represents a conundrum for clinicians. Is this psychosis related to the virus or is it not? And we published a case report in Biological Psychiatry where a 30-year-old man presented with acute onset psychosis at the time of his COVID-19 infection. He had mild COVID-19 that remitted without treatment, but he went on to have persistent and protracted psychosis. He believed that he was talking to God, he was talking to dead ancestors, he became violent, he kicked through a wall, he tried to harm his mother, and he was hospitalized twice without an effective response to antipsychotics. He was unable to work, and after 30 days, the neurologists who had been consulted, Sarada and Lindsay McAlpine, really pushed to treat this patient with immunotherapy because of the temporal association between his acute onset psychosis and his SARS CoV-2 infection. And what they found was that this patient remitted completely within 24 hours and was able to return to work just a few days afterward and has not had a recurrence of his psychotic symptoms. Notably, there was no evidence of inflammation in cerebrospinal fluid or by MRI. But we did immunoassay mouse brain tissue with his cerebrospinal fluid at a 1:4 dilution. And we found that the principally labeled cells by antibodies in his CSF localized to layers 2 and 3 of the anterior medial cerebral cortex. And these were SATB2 positive neurons, suggesting these were callosal projection neurons. And we found that there was evidence of projections within the corpus callosum as well, shown on the right. And so this patient does have an autoantibody to cerebrospinal fluid that binds to a subpopulation of cells that are likely responsible for integration of interhemispheric information. We don't know whether or not this autoantibody predates the SARS-CoV-2 infection, but it was there. We hadn't seen this in over 500 screened samples, but we recently got another biospecimen that labeled the same population of cells in the anteromedial cortex. We don't think that the antigen is the same, but the cell population does seem to be very similar. And this was stain produced by the CSF of a woman in her mid-50s who was admitted to the ICU with delirium, which is an intentional and sometimes psychotic sort of brain disorder in the context of COVID-19. So just a couple more slides here. So I talked to you about data from our first 7 patients and then an 8th patient who's in the case report. We've now collected preliminary data from 44 cases, and we have 46 biospecimens. We have 2 longitudinal samples from 2 of those patients. And I'm just showing you an overview of the characteristics of the data thus far. We have about an equal proportion of males and females, and you'll see that the total numbers vary because we don't have data for all patients in all cases. But we have about an equal proportion of males and females. About a quarter of the biospecimens that have been referred are from the pediatric population. And manuscripts currently under review, but we've identified a bona fide autoantibody in 1 teenage patient. Once again, it binds from the intracellular target. And then nearly an equal proportion of patients who develop neuropsychiatric symptoms during the acute as compared to the post-acute period. Most of the patients have neurologic symptoms, but some patients have both neurologic and psychiatric symptoms, and 5 of the patients have isolated psychiatric symptoms. And more than half of the patients have CSF that stains rodent brain staining. About 3/4 of these patients have detectable SARS-CoV-2 antibodies in their cerebrospinal fluid, and this is consistent with what Carlos Pardo found at Johns Hopkins. And then 7 of these patients, based on the Luminex panel, have antibodies that are unique antibodies to SARS-CoV-2 that are uniquely detected in the CSF and not in the periphery. So some of the conclusions for these early findings is that COVID-19 is associated with acute and post-infectious neuropsychiatric sequelae. That many of the neuropsychiatric COVID-19 patients have anti-SARS-CoV-2 antibodies in their CSF. Some neuropsychiatric COVID-19 patients have anti-neural antibodies in their CSF, and in rare cases, anti-spake antibodies may cross-react with neural tissue. In all of these cases, the pathogenic potential or significance of these antineural antibodies is really unknown and outstanding. And so some of the future directions on the right, I'm actually just showing you a heat map summary of some of the data that I showed you 2 slides below from our expanded cohort. We have 86 total patients now, and so we're continuing to gather data on them. But some of the future directions include, getting some structural affinity data for these cross-reactive monoclonal antibodies, trying to calculate antibody indices. That is, can we determine whether or not the titer for some of these antibodies is higher in the cerebrospinal fluid than the periphery? Are there certain neuropsychiatric signatures? And by that, we really mean, are these autoantibodies really snowflakes? We know based on Aaron Ring's work in the periphery that there's a diverse immune response, and we're seeing that in the periphery as well using our own techniques. And so many of these autoantibodies that we've identified are one-offs, and so these are snowflakes. But might there be subgroups of patients that share an autoantigenic target that may define, a sort of subtype of neuropsychiatric COVID-19? Should we think about panning the exoproteome rather than this linear phage display? Some functional mechanistic studies to ask which of these autoantibodies are functionally impairing. And then we have a study going on here at UCSF, and I think this is going to be perhaps the most important. This is the long-term impact of infection with novel coronavirus COVID-19, and working with Joanna Helmuth. Some patients who have recovered from COVID-19 are incredibly grateful and have offered to undergo lumbar punctures in the absence of a clinical indication for it. So these are patients who don't have neurologic symptoms, don't have psychiatric symptoms, but are willing to donate CSF as a reference control. And then, really thinking about, here we have a specific infection with SARS CoV-2 that temporally we can link to the onset of neurologic and psychiatric symptoms. But, what are the implications? What are the broader implications for neuropsychiatric illness in general? And so I tried to thank many of the people who were principally involved along the way. Of course, my mentors, Sam Pleasure and Michael Wilson, who've just been phenomenal throughout this process. Colin Zemechnik, who's been developing a lot of the tools that have been integral to these studies. Ravi Dandekar, who helped me develop the bioinformatic pipeline for our phage display analysis of the human peptidome. Shelley Farhadian and Eric Song at Yale University. For the mouse model and donating samples. And then, of course, Christopher Barnes at Caltech, soon Stanford, and then Joe DeRisi and Wesley Wu and John Pack of the BioHub, and of course, HHMI. Thank you.
All right, Chris, that was super exciting. Lot to chew on. Audience, remember, go ahead and type your questions into the Q&A box and or raise your hand, and we will call on you. Chris, I want to start off with a bit of a technical question on benchmarking the FIPSeq phage library. So how consistent is the sequences that you get from panning against the FIPSeq library against proteins that you pull down from tissue? Is there a pretty good correspondence between those two?
Yeah, it's a good question. So basically comparing FIPSeq to immunoprecipitation. Mm-hmm. Yeah, so we find that there's not great concordance between the two, and we think that has to do with the sort of biochemical nature of the two different approaches. So with with immunoprecipitation, we use a relatively gentle lysis buffer, Triton, sort of non-ionic detergent. We get out large protein complexes, and we're typically, enriching for conformational epitopes and perhaps post-translationally modified epitopes. Some of the linear epitopes that may be targeted by autoantibodies are probably going to be buried and obscured within the interior of the proteins or at the protein-protein interfaces. And then, of course, by phage display, a lot of these, the epitopes are exposed. We've broken up the proteins, and so we're able to identify a different population of autoantibodies. In some cases, we do see concordance. So, we've used this approach to, this is not my work, but another postdoc who identified a novel autoantibody to a protein called Couch-like 11, and we're able to detect that by both phage and immunoprecipitation mass spec. But, if we search for known autoantibodies that target conformational epitopes, like say antibodies that target the extracellular loop of the NMDA receptor. We can detect them by immunoprecipitation, but not by phage display, because those conformational epitopes are poorly represented in the phage display.
Yeah. But you do think the FIP-seq technique is maybe good for maybe getting a candidate list?
Yeah, I think it's good for getting a candidate list. So once again with the IPMS, it's hard. You get out protein complexes. We've done this with multiple sclerosis, monoclonal antibodies derived from those patients, and have been able to validate some of the hits that come out. But it's a slog trying to wade through that candidate list. With FIP-seq, you're getting individual candidates that were presumably enriched, directly by an antibody. So it's good for generating those lists. I think we've improved our bioinformatic approach. So now our sort of positivity yield is going up. So when we look at a candidate list, we're more confident about what's going to validate. But I think one of the huge advantages of FIP-seq is that because of redundancy within the proteome and then redundancy within the library, we can do multiple sequence alignment to really hone in on a short sequence that we think is targeted by that. We can do epitope mapping, so it's submolecular mapping. And so, just like in genomics, where the pathogenicity of a mutation is going to depend on where it lies within the gene. That may be true for these autoantibodies as well. Some may bind to portions of the protein where there's no functional consequence, and some others may bind to another portion of the protein where there is a functional consequence. And so being able to sort of map those epitopes is going to be incredibly important. We've been able to demonstrate that works using a case where we identified antibodies to a protein called ankyrin G, which defines the axon initial segment, which is where action potentials are generated in neurons. And we found there were a few peptides that overlapped that mapped about a 20 amino acid sequence. We immunostained mouse brain tissue with and without a blocking peptide that encoded those 20 amino acids, and were able to eliminate the immunostaining pattern. So in some cases, FIP-seq gives us the exact target. But I do think that approaches like Aaron Ring's approach are going to be, necessary to expand, the body of autoantibodies that we're able to detect. And I think it was really clever of him to express, full length or longer length proteins using this yeast library.
Yeah. Okay, Sarada's got one now.
Great. Chris, I have a basic question. So looks like these viral proteins are triggering some cross-reactive autoantibodies, which are inducing these neuropsychiatric symptoms, right? So now coronaviruses are respiratory viruses. So is it common for respiratory viruses to cause these neurological symptoms, or is it something specific to SARS CoV-2?
Yeah, it's a good question. It doesn't seem that of course we don't know, but it doesn't seem that the other, seasonal coronaviruses are linked to, acute or subacute onset neuropsychiatric symptoms. Seems to be something unique about SARS-CoV-2 and SARS-CoV-1 within the human coronavirus family. And there is some basic data to suggest that, these viruses directly interfere, like the non-structural protein 13 directly interferes with mounting an immune response. I'm not, uniquely familiar with all the details there, but there are some examples where respiratory infections can lead to a new neuropsychiatric disorder. And so I think that the best evidence for this in terms of not a neurologic disorder, but neuropsychiatric would be influenza and narcolepsy. And so influenza is an incredibly common infection, of course. And so not everybody who is infected with influenza develops narcolepsy. So there seems to be some host susceptibility, which is probably, at least with the influenza association, there seems to be a haplotype based HLA haplotype based susceptibility that predisposes someone to developing narcolepsy after influenza infection. And so perhaps something similar is going on with SARS-CoV-2.
So a related question. So have you compared a scenario that is an autoantibody induced neuropsychiatric disorder in a non-COVID patient? I guess my question is, are there any COVID specific signature in this case?
Yeah, it's a good question. So, what we see in COVID is So with the typical autoantibody-mediated neurologic disorders, they're sort of dominated by a single autoantibody, and so that names the disease: anti-NMDA receptor, anti-AMPA receptor. What we're seeing in COVID is similar to what we saw, what Aaron Ring saw, which is just sort of like a diffuse increase in autoreactivity. And based on our IPMS data, many of these autoantibodies seem to be low affinity. And so there's just sort of this broad autoreactive response rather than a sort of a signature that we've been able to pull out yet. I think now that we're collecting more samples, we're going to be able to look for signatures among subpopulations of these patients. And we certainly don't think that the autoimmunity explains all neuropsychiatric impairment within COVID-19. We think it's going to be a subset, and then a subset of those may have a shared autoimmune signature. But we're going to need more numbers to tease that out.
Thank you.
Chris, okay, a slightly crazier question, trying to get at function of these antibodies. Do you guys have enough to inject into mice and see if you can get some sort of phenotype?
Yeah, that's a great question. So if we can make a recombinant monoclonal, then yes. The bulk IgG injections are a little bit harder to interpret. CSF typically has very low IgG levels. So maybe enough to isolate to inject into a mouse, but it would probably prevent us from doing some of the other studies we would like to do. So the goal, from our perspective, is really to identify an autoantigen in the bulk CSF to clone a population of monoclonals, identify those that target the same antigen, and then use that pure population for functional studies. And so we've done this with two antibodies cloned from a patient with anti-NMDA receptor encephalitis. And so those antibodies were generated in the Wilson lab, and then over in the Pleasure lab, we injected those into mice, and we found it'll be submitted soon. There is some data that's been published on this, but that these antibodies actually interfere with axon guidance of commissural axons. So axons going from somatosensory cortex on one hemisphere to the other hemisphere. So there is a functional readout of antibody exposure. So we'd like to, use that paradigm to test, the functional consequences of some of these other autoantibodies, but, generating them is really sort of the bottleneck right now. But very fortunate, the Foundation for the NIH recently, awarded, funding to me to do exactly that, to clone monoclonal antibodies from neuropsychiatric patients to test function.
That's super exciting. Brian.
Thanks so much for a really amazing talk. I just have a really naive question. Is it understood why auto-antibody production may be induced by infection, but not so much by vaccination, or rarely, or super rarely, or not at all?
Yeah, it's a good question. So I don't think that we exactly know why autoantibody production is being induced by the infection and not being produced by vaccination. Of course, depending on the vaccine, you're being exposed to different components of the virus. So with the mRNA vaccines, you're just being exposed to the spike protein, whereas with some of the adenoviruses, you may be exposed to more of the virus. Certainly with the adenoviruses, we have seen some autoimmunity, and specifically there have been autoantibodies that target platelet factor 4 that predispose patients to clotting and thromboses. And so I think it probably depends on how much of the virus is represented, the route of infection, and whether or not there's an adjuvant with it. But I'm not exactly sure.
Thank you so much.
Yeah.
Thanks, Chris. Chris, going back to the data where you show certain neuronal population cross-reacting for those antibodies, right? So regarding the cortical neurons, regarding the cortical neurons, do we know which? Do we know more about these neurons? Which layers are they in, or are they excitatory, inhibitory?
Yeah. So these are in layers 2 and 3. to the anteromedial cortex, mostly layer 2. There are some other neurons that are scattered about, rare neurons in the hilus of the hippocampus, rare neurons in the thalamus, and some of those other anatomic regions are shown in this case report. But these ones I'm showing here in the cortex are layer 2-3 anterior cortex, and these are excitatory neurons, at least based on morphology and position and SAPE2 positivity.
Great. And this a follow-up question, did you notice any change in the morphology of these neurons, or did they?
That would be a great question that we'd love to answer. These are fixed and perfused mouse brain sections, right? And so these haven't been exposed to, we haven't done like organotypic live, brain slice culture incubated with patient CSF, but that would be something that would be worth looking at to, try to get electrophysiological readouts of pathogenicity or even sort of morphological changes.
Yeah, because it'll be interesting to know if even these neurons have changed functionally.
Yeah, I think that's sort of the difficulty of presenting this work. It's tantalizing, but then, really the question is, is this functionally relevant? And that's what we're hoping to get to.
Thank you. Loren, you go ahead.
Yeah, Chris. So I had a question. What do you think of the more old school approaches at trying to find molecular mimicry? Essentially doing the blast and looking for hits. Of course, there was the high profile disaster of the proposal of anti-syncytin-1 antibodies, leading to the notion that if you get vaccinated, you'll be sterilized, etc.
Yeah.
But I noticed that there are, no shortage of papers out there of people doing blast alignments and proposing antigens. Are any of those worth the paper they're printed on?
So without rendering a verdict on any of those specific papers, I'll just say that, early on, I looked through some of them, and we looked through our FIPSeq data to see whether or not, we could see enrichments, and we did not see enrichments of those proteins with, sequence similarity. That said, I didn't show it today just because of time, but we did do a multiple sequence alignment of the FIP1L1 peptides that were enriched by phage display, and then compared those to the sequence that's targeted on the S2 subunit, just although the spike protein, and there is some sequence similarity, but more interestingly, there's some biochemical similarity. And so we're working with Christopher Barnes to purify the peptide so that we can interrogate the sort of structural relationship between that monoclonal and that peptide derived from SARS CoV-2 spike and FIP1L1. And I think that will give us more insight into the nature of the molecular mimicry. Is there some, so even though, the sequence seems to be short, is there some sort of conformational relationship there, symmetry that we're seeing, or is the antibody, basically adopting a different shape to accommodate these two different antigens? We just don't know, but I think that's going to be something that's really exciting, that we hope to get to soon.
Yeah. Okay. Well, Chris, this was super exciting. I'm super impressed. I'm excited for the future directions. Thank you for your time. Matt, can you bring up the slide for next week, please?
Thank you, guys.
Yeah, we're so excited to have you. Okay, join us next week. We'll have another Double Trouble talk. Chloe Villani and Alex Shalek. So same time, same place, and everyone stay safe. Okay, bye-bye.